Good afternoon, everyone. My name is Enzo Florendo. I'm an incoming junior at Azusa Pacific University, and over the summer, I had the privilege of conducting undergraduate research under the guidance of Dr. Chandra Vulcan Castro, as well as Dr. Rudolph Marcus here on Caltech's campus. And for the past 10 weeks, we have been analyzing the biological processes of F1-ATP synthase in order to determine its chemical states and behavior. So to start off, I'm going to go ahead and give a brief roadmap about what this presentation is going to be like. And so we're going to start off first with the introduction and background of what ATP is, what it does, and some previous research up to this point. Next, we're going to talk about the obstacles, methods, and results. Afterwards, we're going to talk about the relevance and the importance of this research. Then we're going to talk about future steps. And lastly, I'm going to make some acknowledgements before my conclusion. So let's dive right into the introduction and background. ATP synthase is the enzyme in each and every cell which produces ATP molecules. ATP stands for adenosine triphosphate, and it essentially serves as the energy and fuel of your cells. The enzyme itself is divided into two main sections. We have F1 and FO or F0. Those terms are interchangeable. And as you can see in the diagram, F1 is a segment that's circled, and FO is everything else. The scope of my research mainly focused on F1, so that's what I will be talking about today. And that part consists of 3 alpha beta dimers and a rotary shaft, which is the gamma unit in green, as we can see from the bottom diagram. If you look at the way that the alpha beta segments are separated, you can see that they're sort of paired off into each other. So there's 3 pairs. So it makes a hexamer sort of ring, which is attached to the shaft, and it causes it to spin around, and it rotates unidirectionally counterclockwise. Now, I know some of you might be looking at the diagram and you see that the arrow is pointing clockwise, but when the data is actually being gathered, it's flipped upside down, so it would be rotating counterclockwise. So the general movement pattern of the enzyme is that it's separated into what we call dwell and transition states. In short, in a dwell state, it'll be staying in one section most of the time, and then in a transition state, that's when it jumps really quickly to the next one. I do have a little video later on in one of the feature slides, which will let you easily visualize what exactly I'm talking about, so don't worry. But we're going to go ahead and continue on with a bit more background. So a team of scientists in Japan, led by Dr. Noji, they are the ones who actually gathered the data, and the species they have analyzed is Paracoccus denitrificans, F1-ATP synthase, or PDF1 for short. And this is essentially a bacterium. And the way they were able to analyze it is by using single molecule microscopy, which is a somewhat relatively new advancement in microscopy, which allows the user to see individual molecules in proteins. So that is how they were able to do that. Now, as I mentioned, there were some dwell states. In each revolution, there are 3 dwell states. Each one, since a revolution is 360 degrees, each one would thus be 120 degrees apart. Then because of that, we notice that the enzyme exhibits a chemomechanical behavior, meaning that there is a chemical element as well as a mechanical element. And the chemical element being the chemical reaction which causes the mechanical movement. And so those work together to create this behavioral pattern. And the way they were actually able to gather the data is by attaching this 40 nanometer microprobe onto the shaft elastically of the enzyme. And then they put it in a fluid, and they put it in a microscope, and they were able to track the probe as the enzyme spun around. And so they were able to give us that data. But some of you, like myself, might be visual learners, and I know I needed to actually see it in order to see what was going on. And so towards the beginning of the program, I was able to create this code in MATLAB, which is a software that we use to create the plots and the diagrams. And I was able to plot out the movement of the probe. And so I'm going to go ahead and play a bit of it right now. As you can see, right now it's in a dwell state. It's because you can see all the points are sort of staying in that one sector before it quickly goes on to the next one. And then it'll stay there for a little bit before coming back. And then it'll stay there for a bit, and then it'll move to the top right one right there. And so as the graph goes on, you'll notice that these 3 plots start to form, and they create 3 blobs, and they're connected by quite a few, not that many, not nearly as many points, but there are transition points in between. And then the giant cluster of points would be the dwell state. Now, one thing I'm hoping that this graph points out and that you'll notice is that each of the dwell states lasts a different length. This is something interesting that we will be looking at and I will be showing more of in a future slide. But I'm not going to let this play out for too long because in the next slide, I actually have the completed diagram. And we can see 3 units start to form, 3 sectors. Now, each of those would be sort of the dwells, and then in the middle, in between, connecting would be the transition points. Now, if I were to rotate this image 120 degrees, clockwise, it wouldn't be exactly the same, but it would have a general close enough shape where it would look symmetric. Now, that's the problem because in an ideal perfect world, it should be exactly the same. It should be, each blob should be perfect. The transition points should be completely symmetrical to the point where if I were to rotate it 120 degrees, you wouldn't be able to tell. So the reason it's so not perfect and so messed up is some external factors, which I'm going to go ahead and dive into in one of the coming slides. But briefly, this graph over here on the right is what we would call a typical trajectory. Now, is simply a graph of cumulative angle versus time, cumulative angle being the total amount of degrees the probe has rotated, and time being time in seconds. As we can see, this graph goes from 0 to 3 seconds. So the entire data collection process is actually rather quick, but if we zoom in, you can start to see some actual individual points. Each individual point from one to the next, there's 100 microsecond gap, which means that each data set, each complete trajectory has tens of thousands of points. And here, with the naked eye, we can see a staircase pattern form. We can see the dwell states and we can see the transition. Like if I were to select this bottom portion of the graph, you can see that, okay, that's a dwell state because it stays in that one section for an extended period of time. But then the points in between would be transition points. And part of the job was manually determining which points are transition points and which points would then be dwell points. This has to be done manually for now because automating it is actually really difficult. For now, we do have a set criteria which we follow in order to allow for consistency among all the data sets. And as we can see, there are some issues right off the bat that we can see that might be problematic for a computer. For example, this tiny little set of points right there, it's bulky enough to the point where it could be a dwell, but some computers, or depending on how you program it, it might think it's a transition. So one way that we can tell that it is a transition is that, or sorry, that it is a dwell is that, as I mentioned earlier, each of the dwells are separated by 120 degrees. And so if we look at the bottom dwell right here, if we were to draw a line right through the middle, that line would be about 780 degrees. But if we add 120 to that to the next dwell, it would be around 900, which is pretty much exactly where this set of small points is located, which would thus classify this as sort of just a mini dwell. And then 120 degrees higher than that, we get to the next dwell. That would be at around the 10-20 degree average, which as we can see is right where the next dwell is. And so all that lines up. And so part of it is determining, okay, how come these dwells are so short? How come not all the dwells are exactly the same length? If it's perfect and if we live in a perfect world, everything should be symmetric, everything should be calculated and smooth. But here we can clearly see there's a disparity between the dwells. They're not even all the same length. They don't jump exactly the same height. So that is part of the issue. And next, I'm going to go ahead and talk about some of the obstacles which could cause that. And the first one, the most important one, I think, would be Brownian motion. So to briefly summarize what Brownian motion is, it's essentially the colliding and the random particle movement of molecules at the microscopic level when they're suspended in a fluid. And so they're sporadically sort of bouncing off each other and moving a lot, which would cause a disturbance in the data collection, whether the enzyme is bouncing into other molecules, which would cause it to sway a little bit. Maybe it would cause it to prematurely transition, and then maybe the dwell should have lasted a little bit longer. But Brownian motion is an issue that we're looking into. The other issue is the fact that, as I mentioned earlier, it was a 40 nanometer probe, but the system as a whole, the rest of it is roughly 4 nanometers. And so the size difference in that would cause it to, instead of, gracefully rotating around, it might, be a little clunky and robotic in a sense in the movement of the enzyme because it's sort of dragging this probe around. And so that is another issue which we're looking to develop algorithms to mitigate. And a few other ones are rotational and tilt correction. Especially at the nano level, the surface of which the data is being recorded is not going to be 100% smooth. There might be a little bump, which might cause some imbalance somewhere in the trajectory. But currently, one of my colleagues, Nathan Suter, is working on developing algorithms in order to mitigate the effect of this on our data. And so in doing all of this, we're ultimately seeking uniformity, a sense of consistency with the hopes of being able to automate all of this in the future. But the end goal overall is to simply just clarify and elucidate the behavior of the enzyme and trying to understand the chemical reactions and the chemical behavior of why it's doing, what it's doing. So that sort of brings me into the relevance and the importance of my talk. The first, you might be thinking, why is this important at all? It's happening at such a small scale. Like, why would simply understanding it help in any way? But, understanding the behavior of the enzyme is the very first step into potentially learning how to control and manipulate the enzyme in the future. If we can understand how it works, we can potentially maybe even stop the production of ATP molecules in any cell. So let's say, for example, that our target cell is a cancer cell. And if we want to kill it and we find a way to stop the ATP synthase from producing ATP, to stop producing fuel and energy for the cell to live, it would kill it. And so this in the future could potentially be one possible way to combat cancer, which is pretty exciting. The second important thing is that the system, the F1 motor, as I mentioned, it is a biological motor. It's a living, it's a living mechanism. And this motor is actually unbelievably energy efficient. If we were to compare it to a motor you guys might know, for example, like a car motor or a car engine, not electric, but like a general gasoline car engine, those are only like 15 to 25, maybe 30, depending on the car, percent energy efficient, which means only that amount of energy is actually being used towards mechanical movement and generating, movement. But the F1 motor is nearly 100% energy efficient, which means almost all of its energy that it's producing is being used to make something move. So if we can find a way to harness or control or maybe take advantage of this, we can essentially use that to deliver certain materials to certain parts of the body which might not be easily accessible. I believe this is already something that's being produced by other people in the field. I believe it's called microswimmers. Basically, it allows you to inject, a payload. Let's say a certain part of your body or a certain group of cells needs a specific drug. We can put that on and have the biological motors sort of carry it throughout your body until it gets to wherever it needs to go. So that is one possible application in the future, which I would be really excited if it actually worked. So briefly, we're going to go ahead and talk about the future steps. I am happy to say that I will be continuing working on this project throughout the academic school year under the same leader. So the first and most important thing that I hope to be doing is to analyze new sets of data. Now, each data set, there's so much you can do with it. They give you like a spreadsheet of data, and then you can do whatever you want with it. You can plot, time versus cumulative angle. You can plot, position. You can extract the velocity. There's a lot of different angles you can take when it comes to analyzing this data that we've only begun to scratch the surface with all the data sets that we have. So I'm hoping to apply some methods to newer sets of data. Next is working on polishing and refining the correctional algorithms. Some of these are already being implemented, as I said, by Nathan Suter. He's the one responsible for actually generating them. And they help perfect the graph and try and make it so that the effects of these external variables, which are so hard to control, in the outside world, are mitigated. And lastly, simply just continuing to attempt to automate the identification of transition points and hopefully being able to speed up that process, since manually doing it by hand actually does take quite a bit of time. So if we can automate that, that would just expedite our process significantly. So that pretty much brings me to the end of my speech. I would love to actually make some acknowledgments before I finish. First and foremost, Dr. Shandor Volkan-Cascio, who actually, fun fact, is my physics professor at my home institution, Azusa. And he's the one who actually emailed me about this program, and he thought it would be a great opportunity. So he's the one who introduced me to Amgen and SURF and Caltech and research as a whole. So I'm very grateful for him for making this summer possible. Dr. Rudolf Marcus, a Nobel Prize laureate for chemistry. It was an absolute joy to listen to him speak in our Zoom meetings, and he's so passionate about what he does, but it's been really inspirational. Mr. Nathan Studer, former Azusa student and my friend. He is the one, as I said a little bit ago, making the correctional algorithms, and he's the one who helped sort of ease me into this whole project. And lastly, my fellow Amgen scholars, thank you guys for making the past 10 weeks truly amazing, whether it was, weekly lunches or trivia night. This has been an experience I will actually never forget. So that brings me to the end. I do have a Q&A, I believe at 3 o'clock later today. So if you have any questions or comments about anything I brought up today, feel free to leave that comment later on. Thank you very much.